This course spends quite a bit of time on water relations, and there's a couple reasons for that. First is that water relations is one of the things that sets trees apart from other plants. Just the factor of trees' size imposes some interesting limitations with regard to water that don't necessarily exist in other plants. So for instance, the xylem hydraulics of trees ends up being really important, and that's studied a lot in trees and less so in other plants. So that's one reason. And the other reason is just that water is really important to trees and plants. So why is that? What are the reasons that water is important? Well, there's several, and some of these apply not only to plants but to other organisms as well. But um, the the first is really that water is the medium for biochemistry. When we talk about a biochemical reaction like the Calvin cycle, for instance, that takes place in the stroma of the chloroplast, that's happening in a water solution. So clearly water is important for almost all life, and that's one of the reasons for that. In plants and trees, it's also really important that water is the medium for transport. When substances are moved around in the tree, they're moved around in solution, whether it's in the xylem or the phloem. Those constituents are moving around in a solution of water. So clearly that's an important piece as well. Another is that turgor pressure provides support for non-lignified tissue. And we'll define turgor pressure in a little bit, but suffice it to say, if you think about a plant cell, turgor pressure is just the pressure pushing out from inside the cell on the cell wall. And in herbaceous plants or in non-lignified or less heavily lignified parts of trees, that pressure is actually what gives rigidity to leaves and non-lignified stems. So that turgor pressure can be important for that reason. Another is just that vast quantities of water are released by plants essentially as an unavoidable cost of opening, well, it should be of opening stomata. So in C3 and C4 plants, the stomata need to be open during the day to allow CO2 to diffuse in for photosynthesis. But as a consequence of that, the moist interior of the leaf is exposed to the drier air and water evaporates. So at the same time that CO2 is diffusing in and being used in photosynthesis, water is evaporating. And that's a cost, but it's unavoidable. We've talked about some workarounds. CAM is a workaround, photosynthetic pathway to that, but really most plants and all trees have to lose a lot of water. Probably 100 molecules of water are lost for every molecule of CO2 that's fixed by photosynthesis. So it's an inevitable and unavoidable cost. So plants use a lot of water. And the final, and this is sort of related to D, but that's that there's a strong relationship between water availability and productivity. In other words, the growth of plants tends to be pretty dependent on whether there's abundant water supply in the soil. This graph demonstrates that. Now this is pretty old, and I think there's probably some updated graphs with more data, but this relationship generally holds in that production, and this net primary productivity is just a measure of plant productivity or plant growth. When that's plotted against some measure of water availability, in this case, this is annual precipitation in different locations, there tends to be a pretty strong relationship between the amount of water available and basically how much growth can occur in that ecosystem composed of plants. Now there's a lot of scatter around that line. So for instance, at 1500 millimeters, you can see there's, a more than twofold variation in productivity. So think about what some of the other factors might be, for instance, that differentiates the productivity between that point and that point that have the same essentially water availability in terms of precipitation. One of those might be the amount of evaporation. It could be that, for instance, this location is hotter, so it has more evaporation from the soil during the year, so there's less water available for plants. It could also be, could be more evap. It could be cooler temperatures. So for instance, it could be that that site receives 1500 millimeters of precipitation, but the annual temperature is relatively cold. So growth is limited by temperature rather than by water in that particular location. There could be situations where soils limit growth. So certainly there are factors other than just water that limit plant productivity, but certainly water is an important one. And when we look at where, for instance, continuous canopy forests occur, the green places on this map, those are generally associated with precipitation. So if we look, for instance, this large unforested region in the middle of the country, that's the Great Plains. And then these regions in the west, that's, the Great Basin, the Great Western Deserts. So that variation broadly is associated strongly with precipitation or water availability. So for instance, this map shows variation in annual precipitation in the contiguous US, and you can see there's pretty strong gradient in precipitation from east to west. It gets drier and drier as you go west. And if you remember, it was sort of this region here where there are no trees, and this region here where there are no trees. Great Plains here, Great Plains, and then desert here. The green, the spots of greater precipitation here, these are on the windward sides of mountains, so that as bodies of air reach the mountains and rise, they get cooler and drop rain. So you actually will find forests in some of those locations because there's more precipitation. Another point to make, for instance, where I grew up in Oklahoma City here, there's no continuous canopy forests, and precipitation there is probably, this is in inches, maybe 36 inches of precip. Well, where I went to graduate school, is right here, Seattle. And Seattle probably gets 36 to 40 inches of precipitation in a year. But Seattle's called the Emerald City. It's heavily forested. So the difference there is that evaporation component. Oklahoma City has really hot summers, so that lessens soil water availability, while Seattle, the Pacific Northwest, very mild summers, although it doesn't rain a lot in the summer, there's also not as much evaporation. So certainly the determinant of water availability is not necessarily absolute precipitation, but the balance of precip minus, sometimes it's called actual evapotranspiration. So that's an index of water availability. So really that's the important factor. But at any rate, precipitation broadly drives patterns of forests. Now, again, there are other things. Soils can have an impact, but also disturbance. So for instance, burning, both lightning ignitions, but also human burning in the Great Plains is one of the reasons there's not continuous forests there. So in areas, for instance, in here, that are where burning is excluded, you actually start seeing encroachment of woody vegetation into prairie. So as in always in ecology, it's never one factor. There's always interacting factors. Another example of the importance of water for trees and forests, this is a demonstration of aspect. And I always like to show this to my students in Florida because we don't get many examples of aspect in Florida. But this is a nice example driving east on I-90 out of Missoula, Montana. And I'm heading east, so that slope on the right is facing north, and the slope on the south is facing, on the left is facing south. And there's a big difference in tree cover on those two slopes. There's continuous tree cover on the north-facing slope, or the north aspect, and not continuous tree cover on the south aspect. And that's because of those are differences in water availability. That south-facing slope has more sun exposure, more evaporation, and therefore less water availability to trees. And that drives differences in vegetation over, in this case, a couple hundred yards of distance. So clearly the climate is the same, but the local soil conditions are different due to differences in exposure to the sun on north and south facing slopes. And so in mountainous regions, aspect can be a really important factor that plays into water availability and vegetation on those slopes. The central concept for talking about water relations in soil, plants, and atmosphere, sometimes it's called the soil plant atmosphere continuum, is sometimes what you'll see that referred to, is water potential. Water potential, it can be defined as the potential energy of water. It's usually per unit volume, but the units are not explicitly per unit volume, but potential energy of water relative to a standard state of water. And the standard is pure water, so water with nothing dissolved in it, at standard temperature and pressure. So that's the reference. So water at standard temperature and pressure, which is basically 20 degrees C at sea level atmospheric pressure. So you can think of a beaker of pure water sitting on the lab bench here in Florida. That's almost standard temperature and pressure at room temperature. That has, by definition, a water potential of 0. So all other water potentials are expressed relative to that. So generally, as water is bound, so I sometimes think of it as water being bound by solutes or being in a plant cell, for instance, its potential is reduced relative to standard. This means most water potentials are negative. Because we're seeing a reduction from 0. This can be confusing thinking in a negative scale. So when we say, for instance, water potential is reduced, what that means, when I say, and I try not to say it this way, but if you hear someone say water potential is reduced, that means water potential is more negative. And that can be confusing because that is a bigger negative number, so the absolute value of that number is bigger, but it's a reduction. So I usually try to say more negative or less negative when I'm talking about relative values of water potential. That, I think, can help clarify a little bit. For the most part, water potential is the standard in the literature for thinking about the movement of water in plants. There are some exceptions. In your notes, there's a list of references, Sinclair and Ludlow, Kramer, Paciora, Schulze et al., and so on, where there was an argument in the literature about whether water potential is indeed the best measurement of water status of plants. A lot of that was initiated by Tom Sinclair, who was an agricultural research service researcher here in Gainesville, Florida. And Tom, I was able to work with Tom over the years, and he served on some of my students' committees. Tom's a brilliant man and really interesting guy, fun to work with, but he did like to stir the pot. And Tom, I don't think, ever sort of settled for the idea that water potential was the best measurement of water status of plants. He really thought that other measurements, like water content, were actually better indicators of water status of plants. So take a look at those papers. And remember good old Dr. Sinclair. He's retired now up in North Carolina with his grandkids. But Tom always liked people to think critically about concepts out there. The framework we usually use for water potential is where a total water potential is the sum of several component water potentials. So in this case, and psi is the Greek letter that's usually used for water potential. In this case, total psi water is just the total water potential at some point in the plant or soil. Usually when we talk about this, we say, okay, in leaf xylem. The water potential, total water potential is this, something like that. So specifying where that is usually helpful. Psi pi, this is osmotic potential. Okay. Osmotic potential is just a reduction in water potential caused by the dissolution of solutes in water. Okay. So as a solute, say sugar or salt, is dissolved in, let's think about our beaker of water. It's pure water at standard temperature and pressure that has a water potential of 0. If we put a spoonful of sugar in there and mix it up, as those sugar molecules dissolve, they bind some of the water molecules in that water, and the water potential of that water will go below 0. It will become negative. So the osmotic potential component is always 0 or negative. It's never positive. And I also meant to note that the total water potential in plants is usually a negative value. There are a few situations where you actually might have a total water potential of 0, but that is rare. Fully turgid cells really is where that happens. Psi p is pressure potential. Psi p is a little confusing because it basically has two separate functions, or it signifies two main conditions in the plant. One is in living cells, so say a mesophyll cell in a leaf. This is just turgor pressure. So I've mentioned turgor pressure. Turgor pressure just refers to the pressure exerted on the cell walls by the fluid inside the cell. I sometimes think of a plant cell using a cartoon model where a plant cell is like a water balloon inside a cardboard box. And when that water balloon is fully inflated with water, that the balloon itself is the cell membrane, the cardboard box is the cell wall, and when that balloon is fully inflated, it's pressing out against those cell walls, and the balloon is making contact with the cell walls all around. That pressure between the water balloon and the box is turgor pressure. Okay. Now in tracheids and vessels, psi p is usually negative. So in dead water conducting cells like tracheids and vessel elements, psi p, the value of psi p is usually negative. And I'll elaborate on negative pressures in a little bit. Negative pressures are really weird, actually. But I'll elaborate on that in a little bit. For now, just know that psi p can be either positive or negative. It's positive in living cells. It's negative in dead water conducting cells like tracheids or vessel elements. And then the final element is psi g. That's gravitational potential. That's just a change in energy status of water due to its position relative to some reference plane like the ground. Usually that's the ground. So this is the force of gravity on water. So if we have that beaker of water at standard temperature and pressure, that has a water potential of 0. If we raise that beaker of water a meter above the lab bench, it now has a different energy status because it's been lifted above that reference plane of the lab bench. And the gravitational potential is just a function of height. Gravitational potential is plus 0.1 megapascals per 10 meters of height. Now, when you look at the literature on water potential in soils and in agronomic plants, for instance, you'll seldom see reference to gravitational potential because this effect is very small over the distances that we're talking about in soils and in short agronomic plants. But in trees, this can actually become a fairly substantial effect. So in a 100-meter tall redwood, the force of psi g alone is 0.1 times 100 meters equals 1.0 megapascals, just due to the effects of gravity. So, in other words, lifting water to the top of a tall redwood requires significant force, 1 megapascal of pulling force just to pull the water that far. And I should have mentioned this earlier, but the standard units for water potential are megapascals. In old literature, sometimes you'll see, and these are pressure units. So in some old literature, you'll see references to bar. So 1 bar equals approximately 0.1 megapascals. Okay? This graph shows some data from a study where folks measured water potential at different locations in a Douglas fir tree. These were all taken pre-dawn, so what that means is that there's no transpiration. And these, actually the leaves were covered with aluminum foil as well, so there's no effects of transpiration. So really what you're seeing here is mainly, what we're mainly seeing is the effects of soil water potential and gravitational water potential. So here at the base, basically they measured an understory tree that was not transpiring, and it's in relative equilibrium with the soil. So it's at negative 0.3 megapascals. So that's essentially the water potential of the soil. That's a good measurement. And then they measure water potential going up into the crown of the tree. so at different heights. And they've plotted a couple of things. One is this solid black line is the theoretical gravitational potential. So this has a slope of 0.1 megapascals per 10 meters, or 0.01 megapascals per meter. So what you can see is that black line. So if we start at negative 0.3, negative 0.3 megapascals, and we go up 20 meters, so 20 meters, 20 times 0.1. Actually, since it's per 10 meters, that should be 2 times 0.1 equals 0.2. So the force of gravity reduces another 0.2 megapascals, and the essentially gravitational potential at that point, including the soil water potential, should be negative 0.5 megapascals. So that's what that graph shows. And what they showed is that these dots are where they actually measured water potentials at different heights. They went up at approximately this slope, but they were a little slightly more negative than would be predicted just by that hydrostatic gradient due to gravity. They were slightly more negative because there's friction in the system. This is not just a theoretical system. There's friction due to the pulling of this water against the xylem. And so that makes the values a little bit more negative. But, you know, this is a demonstration that gravitational potential can be an important factor in tall trees. You know, we don't have any 20-meter wheat plants, but certainly we have lots of 20-meter trees. And in this case, this is, you know, a 60-meter tall Douglas fir. So gravitational potential can be really important in some of these tall trees. I just want to make a couple of points here at the end of this section that are really important to keep in mind when we're thinking about the interaction of all these factors in the water relations of trees. And the first is that preservation of positive turgor pressure, so that's the psi p component, in living cells is critical. When we think about a living cell, that cell must be turgid. It must have a positive psi p in order to live. And that's because that positive turgor pressure presses that cell membrane against the cell wall and allows that cell to be connected to adjacent cells. If that cell starts losing water, and losing turgor pressure, the cell membrane will pull away from the cell wall and that cell will die. So that plasmolyzed cell, in all but a few special situations, that cell's dead. There's nothing that can be done. So a lot of the water relations responses of plants have evolved around maintaining positive turgor pressure in living cells. So that's an important evolutionary driver and important to keep in mind when we think about stomatal closure, for instance, and the triggers of stomatal closure. Those are generally have evolved around preserving psi p in living cells. The other really important point to keep in mind is that water moves from less negative to more negative water potential. So when we're looking at water flow in a plant, water generally flows from either less negative to more negative psi W or less negative to more negative psi P, depending on which part of the system we're looking at. So water movement is down that gradient from less negative to more negative water potential. Keeping those two things in mind will really help you to understand the significance of these water relations concepts within the context of the plant.